Hello students, my name is Niyati Said and thanks for watching Edupedia Word videos. My topic for the presentation is cell organelle and their function. In this tutorial, we will continue to discuss about endomembrane system, specifically the flow of material between endomembrane system and plasma membrane. Now the question arises, what is cell endomembrane system? So it carries out a variety of tasks in the cell, including protein synthesis and transport of protein into membrane and other organelle. Second, it helps in the movement of lipid. Third, it helps in the detoxification of poison. And fourth, membrane of this system are related either through direct physical continuity or by the transfer of vesicles. Now, what are the parts of endomembrane system? One is plasma membrane and cell wall, inside the nucleus, ribosome, endoplasmic reticulum, Golgi apparatus, lysosomes, vacuoles, mitochondria, chloroplast and cytoskeleton. Now we will discuss this in detail. First is cell boundary. So cell membrane and cell wall lie to this category, that is cell boundary. Cell membrane, it is a very thin flexible membrane that surrounds the cell. It is made up of two layers of phospholipid which is intervened with cholesterol and proteins. See, this is the typical diagrammatic representation of plasma membrane. It is, see, you can see that it is made up of phospholipid bilayer and carbohydrate is embedded in it and proteins are also embedded in it. This is phospholipid, this is the hydrophobic, this is a hydrophilic end of the phospholipid and this is the hydrophilic end of the phospholipid. And now comes the cell wall. Cell wall is a distinguishing feature of plants. It is always present in plants, not in animals. It is a rigid layer around the membrane found only in bacteria, fungi and plants. As you can see that two cells are connected to each other by a middle lamella. Middle lamella is acts like a glue between two cells. Okay. And this yellow part is a typical cell wall. Now come to cell membrane. We'll discuss this in detail. It's a fluid mosaic model 2. So cell membrane is made up of two layers of phospholipid which is intervened with cholesterol and protein, as we have read. And secondly, it separates cell from outside environment and regulates transport of molecule and signal into and out of the cell and communication with other cells too. And secondly, it is the attachment point for the intracellular cytoskeleton. Third, it maintains essential difference between cytosol and the external environment. See, this is the cytosol or cytoplasm, and this is the extracellular matrix. So this plasma membrane, it helps in the maintaining essential difference between cytosol and extracellular environment. So if we summarize cell membrane, then what it does, it provides support and protection. Secondly, it helps in entry and the exit of the molecule. You can say that it's like our school door and intercom system. And always note that one thing, that plasma membrane is about 5 to 8 nanometer in thickness and plasma membrane is selectively permeable. Selectively permeable means it allows only selective molecules to come inside it, and thus it regulates cell molecular traffic. Now we will discuss about the fluid mosaic model of plasma membrane. It was proposed by Singer and Nicholson, which displayed the cell membrane as integration of protein and other molecular in the phospholipid bilayer. Okay. The fluid mosaic model states that a membrane is a fluid structure with a mosaic of various proteins embedded in it. The cytoskeleton is attached to the cell membrane for added stability, since membrane protein and phospholipid can shift places in the membrane. Now we will discuss about the phospholipid and membrane protein in detail. So phospholipid, you can say that they are a structural framework of plasma membrane. Phospholipids are made up of glycerol backbone with a hydrophilic head region containing a phosphate group and hydrophobic tail region containing a saturated fatty acid and unsaturated fatty acid too. There are typically 5 types of phospholipids present. One is phosphatidylcholine, second is phosphatidyl'inositol, third is phosphatidylserine, fourth is ethanolamine, and fifth is sphingomyelin. Phospholipid play an important role in the transport of fat between gut and liver in mammalian digestion. Now we will discuss proteins in detail, membrane proteins in detail. A membrane is a collagen of different proteins embedded in a fluid matrix of the lipid bilayer, as I have shown you just now. Protein determine most of the membrane specific function. There are 2 types of protein. One is integral or intrinsic protein and second is extrinsic protein or peripheral proteins. Integral proteins, they are located in these. Intrinsic proteins are located in lipid bilayer, penetrate the hydrophobic core and often span the membrane and are called as transmembrane protein also. So integral protein, they involve in cell to cell signaling with surface carbohydrate chain, and peripheral protein, they are not embedded in bilayer and they can found on either surface of the bilayers. Always note one thing, that membranes are in motion with the fast drifting lipid and slower drifting proteins. Now we will discuss about diffusion and osmosis in detail. See, this is low concentration area, low sugar concentration area and high water concentration area. And this is a high sugar concentration area and low water concentration area. And this is a selectively permeable membrane. That means that it will allow only specific molecules to enter through it. This blue indicates water, these blue bubbles indicate water, and the green bubbles indicate sugar. Now what is diffusion and what is osmosis? Diffusion states that movement of particle from an area of high concentration to low concentration area. Okay, and osmosis states that water diffusion takes place through a selectively permeable membrane. Water will move into and out of the cell to equalize concentration. If water will move inside the cell, then what will happen? Cell will become turbid. And if cell water moves out of it, then cell becomes flaccid or it becomes shrink. See, we'll first discuss about the osmosis practice. This is a jar and this is a cell which is inside that jar. The concentration of cell is 90% water and 10% NaCl. And concentration of that jar solution is 90% water and 10% NaCl. So tell me students, the concentration is equal. That means cell concentration and jar concentration is equal. Then what will happen? Concentrations are equal. So no net movement of water will take place. Okay. Now we will discuss about the second osmotic practice. The cell concentration is 90% water and 10% NaCl, and solution concentration is 80% water and 20% NaCl. Then what will happen? Concentration of water is greater in the cell than outside. So water will move out move out from the cell. Then what will happen? Cell will become flaccid. That means it will shrink because its water is moved out. Now we will discuss about the osmosis practice. Third, suppose cell concentration is 80% H2O and 20% NaCl, and solution concentration is 90% H2O and 10% NaCl. Then what will happen? Means the solution concentration water level is higher than cell, because 80% water is there in the cell and 90% water is there in the solution. Then what will happen? Water will move in and it will make cell turbid. So as you can see, that concentration of water is greater outside the cell than inside. So water will move in. Now what's the difference between facilitated diffusion and the active transport? Facilitated diffusion, it requires protein channels, that is carrier protein, and they are named as transporters or channels. On the contrary to this, in active transport, it requires a carrier protein and they are named as pumps, protein pumps. Secondly, they are extremely specific in nature, and the same applies with the active transport. They are also extremely specific in nature. Third, they get inhibited through competitive inhibitors. Means when competitive inhibitors come, so they can inhibit their diffusion. Whereas on contrary to this, they are also inhibited through competitive inhibitors. Fifth, solutes are transported from high concentration area to low concentration area. This is the distinguishing factor of facilitated diffusion. Whereas solutes are transported to high concentration from low concentration area. When a solute moves from high to low, then no energy is required in facilitated diffusion. That is obvious. And when solute move from lower concentration to higher concentration area, then it requires energy. So facilitated diffusion, when the plasma membrane helps particle pass through protein channels, no energy is used. And what about active transport? When the plasma membrane uses energy to move particle against the concentration gradient, that is from low concentration region to high concentration region. That's why they require more energy. They uses transport proteins. Now comes nucleus. Nucleus contains DNA, that is a genetic material which makes you who you are now, and it directs the activity of the cell because it is considered as a head of the cell or the brain of the cell. For example, when it grows and divides. Now just think of our head's office as the nucleus, because nucleus is the brain of a cell, and the student files in the head office as deoxyribonucleic acid, that is DNA, our genetic material. See, it's like a principle of our school. Now inside the nucleus, another component of our cell endomembrane system. Nuclear envelope, a membrane that surrounds the nucleus. Second, chromatin. It consists of DNA wrapped around proteins. Means it appears as a ball of string, like in Hindi you can say motiyon ki mala, where ball appears as a shape of protein and a string is a DNA. Chromosomes. Chromosome is a condensed string-like structure that forms from DNA just before the cell divides. And then comes nucleolus, structure within the nucleus that makes ribosomes. This is nuclear envelope, this is chromatin, and this is nucleolus. Now come to ribosomes. Ribosomes are made up of RNA and protein, and they are the construction guys of protein. They are granular structures. They are composed of ribonucleic acid, RNA and protein, and are not surrounded by any membrane. Eukaryotic ribosomes are ATS. ATS represents Swedberg unit, okay, or sedimentation coefficient, while prokaryotic ribosomes are 70S, that it stands for sedimentation coefficient. S, it is indirectly a measure of density and size, you can say. And both 70S and 80S ribosomes are composed of two units, one larger subunit and another small subunit. Okay. So what is the function? You can say that ribosomes are the protein builders or the protein synthesizers of the cell. They are like construction guys who connect one amino acid at a time and build long chains. Now comes endoplasmic reticulum, that is ER. ER was unknown in the days when we were restricted to light microscope. In fact, in those days, the content of the cell were thought to be formless, soupy protoplasm, but electron microscope reveals astounding complex membrane system that takes up a large part of the cytoplasm of eukaryotic cells, especially those cells that are engaged in significant protein synthesis. Lipid, proteins and other products are also made up here. Hence, you can say they are like membranous tunnels, okay, and they are like tiny tubular structures scattered in the cytoplasm, and hence ER divides the intracellular space into two distinct compartments, that is luminal inside the ER and extraluminal, that is cytoplasm compartment. ER appears in two main forms. One is rough ER, which has ribosomes in it, and second is smooth ER, which has no ribosomes on it. ER often shows ribosomes attached to this their outer surface. The ER bearing ribosome on their surface is RER. In absence of ribosome, they appear smooth and are called as SER, that is smooth endoplasmic reticulum. Now, what is the function of RER? RER got its name from the appearance of dense granules that tightly adhere to one side of the membrane. These granules are ribosomes, okay, frequently observed or involved in protein synthesis and secretion. These RER are frequently involved in protein synthesis and secretion also. RER are extensive and continuous with the outer membrane of the nucleus. Now, what is the function of SER? These cells have ribosomes, but they are usually not bound to membranes. Instead, the ribosome float freely in the cytoplasm. See, as you can see that its ribosomes are floating freely on the cytoplasm. It's not attached with the smooth endoplasmic reticulum. So now, what's the function of a smooth endoplasmic reticulum? You can say that it is a major site for synthesis of lipids, okay? And in animal cells, lipid like steroidal hormones are synthesized in SER. It is currently believed that enzymes of lipid and steroid are found in a smooth membrane of endoplasmic reticulum. Now, how vesicles are used to transport materials within a cell between the endoplasmic reticulum, Golgi apparatus and plasma membrane? This question mainly comes in pre-medical tests, that is PMTs. So what happens is, the process of protein synthesis starts when mRNA, that is messenger RNA, moves from the nucleus to a ribosome on the surface of RER, that is rough endoplasmic reticulum. As the ribosome, which are construction guys of protein, as the ribosome builds the amino acid is a monomer unit of protein. The chain is pushed into the cisternal space of the RER. We will discuss what is cisternae but later. When the protein are complete, they collect and RER pinches off a vesicle. The polypeptide is finally transferred via vesicle to the plasma membrane, whereby it is either immediately released or stored for a delayed release in response to some cellular signal. See, as you can see that this is nucleus, this is the nucleus wall, and this is a rough ER, that is rough endoplasmic reticulum. It is always in a close association with the nucleus. These are its cisternae ends of RER. When it pinches off the cisternae, then it becomes vesicle. Now this vesicle moves to the membranous tunnel, that is Golgi complex, and then Golgi complex, it also pinches off a vesicle, and thus this vesicle, as you can see, this vesicle moves through to the plasma membrane, and the polypeptide is finally transferred via vesicle to a plasma membrane, whereby it is immediately released or stored for delayed response response to some cellular signal. Now what are its functions? It helps in intracellular transportation, as you can see, as we have just read, and second, it provides mechanical support to cytoplasmic matrix, and third, it helps in the formation of nuclear membrane and Golgi complex, and it also helps in detoxification of poisons and metabolic waste, and it is the storage house of lipids and carbohydrates. Now comes Golgi apparatus. Golgi apparatus is also known as Golgi bodies. They consist of many flat disc-shaped sacs or cisternae of around you can say 0.5 micrometer to 1 micrometer diameter. Okay, varied number of cisternae are present in Golgi complex. Cisternae are concentrically arranged near the nucleus with distinct convex six, that is forming phase, and concave trans, that is maturing phase. Okay, cis and trans phase are interconnected. Now just think of a lunch faculty who bring cafeteria food into the school and then packages it and stores it in the kitchen for the later use. This is a Golgi body. Golgi apparatus, so what it does, it modifies, it sorts, and it packages proteins. See, this is the trans phase of the cisternae of Golgi apparatus, and this is the cis phase, that is a receiving phase. It is always in a close association with the rough ER. See, this is rough ER. So whatever vesicles or newly synthesized proteins are formed, they come from the vesicle, as we have just read in endomembrane system that it pinches off the vesicle. So this vesicle is containing a newly synthesized protein which goes to the Golgi complex and fuses with it. See, it got fused, and then it goes from the shipping phase. It pinches off from the cisternae and becomes again a vesicle and goes out of the plasma membrane. Why Golgi apparatus remains in close association with the ER? Golgi performs packaging. So materials to be packaged in the form of vesicle from the ER fuses with the cis phase of Golgi apparatus and move towards the maturing phase, that is a shipping phase, as I have just explained. So this explains why Golgi apparatus remains in close association with the endoplasmic reticulum. This is Golgi apparatus. In 1988, Camillo Golgi, an Italian cytologist, discovered that even when he treated the cells with silver salts, certain peculiar bodies showed up in the cytoplasm. The Golgi apparatus is a series of membrane-shaped pancakes. Okay, they are like pancakes. The single membrane is similar to the cell membrane in that it is also made up of bilayers of phospholipid. The membrane surrounds an area of fluid where the complex molecules, that is protein, sugar, enzymes, they are stored and changed, because the Golgi complex absorbs vesicle from the rough ER. Now the foundation of vesicle. This is a typical diagrammatic representation of foundation of vesicles. How vesicles forms? See, this is membrane and receptor is attached with this membrane, and this is a ligand, this is adaptin, and this is clathrin. Okay, the coat protein must have a membrane protein to bind to like a receptor. For clathrin, these are called adaptins. Okay, these are called adaptins. This is a coat protein, and they must have a membrane protein to bind like a receptor. And then clathrin. This clathrin recruits more clathrin and adaptins. Once a bud is formed, see, now the bud has formed. Now once the bud is formed, another mechanism is required to sever it from the membrane and make a vesicle. See, this dynamine comes into the scene and it pinches off the vesicle as soon as the bud forms, and thus vesicle is released. This job is performed by multi-subunit complex called dynamine. Now question. Now what is the function of Golgi body? It performs function of packaging material to be either to the intracellular targets or the secreted outside the body. They produce lysosomes, that are suicidal bags. They secrete various enzymes, hormones, and cell wall material. They help in phragmoplast formation. Phragmoplast is a distinguishing feature of plants. They are always present in plant cell and absent in animal cells. Now come to lysosomes. What are lysosomes? Lysosome is a membrane bound vesicular structure formed by the process of packaging in Golgi apparatus. Okay, they are also known as suicidal bags. Lysosome vesicles have been found to be very rich in almost all type of hydrolytic enzymes. That's why I have compared it with Lysol. It's like a caretaker who cleans up any waste in the school at the end of the day. See, I always relate it with the outer world. It helps in a better understanding, students. Lysosome, so they are small organelles filled with enzymes. As I have said, the enzymes such as lipase, that which break fat and lipid, hydrolases, carbohydrates, and protease. They have found to be you can say very rich in almost all type of hydrolytic enzymes. These enzymes are capable of digesting carbohydrate, protein, lipid, and nucleic acid. See, phosphate ester which help in the formation breaking of the acid phosphate. Nucleases, as its name is suggesting, that it will break DNAs and RNAs. It also has protein digesting enzyme, that is collagenase, cathepsins, and other proteases. And carbohydrate digesting enzymes are also present, such as beta glucosidase, hexosaminidase A, and alpha manosidase, alpha fucosidase, and sialidase, etc. And it also has a lipid digesting enzyme, that is lipase, and for example sphingomyelinase esterase. Okay, lysosomes are like a recycling center that work smoothly until a problem disrupts the operation. That's why they are also known as time bombs. and suicidal bags. Lysosome is responsible for degradation of macromolecules in animal cells. The mature lysosome is filled with so many hydrolytic and acid hydrolases enzymes that are active at very low pH. Always note that it works at a very low pH. These includes nucleases that degrade nucleic acids such as DNAs and RNAs, and proteases, the enzyme that degrade proteins, and glycosidases, the enzyme that degrade oligosaccharides and polysaccharides, and lipase that degrades lipid. Material is delivered to the lysosome via endosomal pathway. Now we will discuss endosomal pathway in detail. An endocytic vesicle fuses with the early endosome. This in turn will mature in late endosome and finally into lysosome. So what are the three stages? First is early endosome, then what it forms, what it forms, late endosomes, and then finally into lysosome. The material delivered to the lysosome will be degraded, and in some cases the breakdown products such as amino acids, nucleotides and sugars will be transported to the cytoplasm and recycled by the cell. This is the endocytic pathway. See, I have told you three stages. One is early endosome, then it matures and forms late endosome, and thus finally it forms lysosome. Okay. An endosome that is destined to transfer its content to the lysosome generally goes through several changes along its way. In its initial form, this early endosome in its initial form, when the structure is often referred to as early endosome, which contains single compartment. Over time, however, chemical changes in the vesicle takes place and membrane surrounding the endosomes folds upon in itself in a way that it is similar to the invagination of a plasma membrane. Then what happens? Then multivesicular endosome is formed, which is an intermediate structure in which further chemical changes, including a significant drop in pH level to the more lower pH, it takes place as a vesicle develops into the late endosome. And through late endosomes, they are capable of breaking down many proteins and fat. A lysosome is can fully digest all the material they contain. Frequently, the content of late endosome are conveyed to the lysosome through fusion of their membranes. So that was all about endocytic pathway. And now, what are the functions of lysosome? They are concerned with intracellular digestion. That is why they are known as suicidal bags or time bombs. They also contribute to aging process. They destroy old and non-functional cells which bear them, and this process is known as autolysis. Lysis means breakdown and auto means the one who does itself. So they are also known as suicidal bags or time bombs. They break worn out cells, damaged cells and cell organelles to component molecules. That's why they are known as suicidal bags. For building new cell organelles, so they are also called as recycling centers. Now come to vacuoles. Vacuoles are storage bubble found in the cell. They are single membrane bound sac like vesicle present in the cytoplasm. Now tell me, what does it contain? It contains sap, excretory product, other material not useful for the cell. Vacuole is always bounded by single membrane called tonoplast. Tonoplast is semi-permeable in nature. That means it will allow only certain molecules to pass in. In plants, tonoplast facilitates transport of a number of ions and other material against concentration gradient into the vacuole. Hence, their concentration is significantly higher in the vacuole than in the cytoplasm. Both cell size and turgor pressure are regulated by the vacuoles. In amoeba, contractile vacuole is important, very important for excretion purpose. Vacuole maintain a much higher ionic concentration than cytoplasm, and as a result, water enters by osmosis. Now, what are the functions? Functions that vacuoles have the facility to contribute to the rigidity of the plant, to cell elongation, and to the processing and storage of waste products. This makes them very unique and and they are distinct organelle in their own right manner. Okay, vacuoles assist with growth of the cell. They help in the cell elongation process. They store nutrients and non-nutrient chemicals. That's why they are also known as storage bags. Voracious vacuole. See, as we know that both the cells, means plant cell and animal cell, they both possess vacuoles. So vacuole in animal, they store waste, and whereas vacuole in plants, they store water. Okay. Now comes mitochondria. Mitochondria is a powerhouse of the cell, as we have read in our previous class. Mitochondria is an organelle that converts chemical energy stored in food into compounds that are more convenient for the cell to use. Okay, it is saucer shaped and or cylindrical, having diameter of 0.2 to 1 micrometer and length is about 1 to 1.4 micrometer. It is a double membrane bound structure with outer membrane and the outer membrane and the inner membrane. Both the membrane divides its lumen distinctly into two aqueous compartments. Inner compartment is called as matrix, that possesses single cellular DNA molecule, and outer membrane forms the continuous limiting boundary of the organelle. Inner membrane forms a number of infolding called as cristae towards the matrix, and cristae helps in the increase of the surface area of the mitochondria. Now the question arises, why it is called as powerhouse? Because they produce cellular energy in the form of ATP. Now just think of a lunchtime at a school. Food is taken out of packages, cooked, and then provided to the students and teachers to recharge them for the rest of the day. That is why this is known as powerhouse of the cell. This is the mitochondria, which is saucers in shape. Now come to chloroplast. Chloroplasts, they are also known as plastids. They are found only in plant animals and euglenoids. They capture energy from sunlight and convert it into chemical energy, and that process is known as photosynthesis. They are found only in plant cells. They bear some pigments like chlorophyll, that thus it imparts green color to the plants. They are large in size, so they can be easily seen under microscope. See, this is the diagrammatic representation of chloroplast. It has two membranes, just like mitochondria, outer membrane and the inner membrane. These sac of coins are called as thylakoids, and this is stroma, this fluid, and singular part of thylakoid is grana. See, this is chloroplast. They are large in size, so they can be easily seen under the microscope, and based on the type of the pigment, plastids can be classified into three: chloroplast, chromoplast and leucoplast. Pigment like chlorophyll, which are responsible for trapping light energy, which is very essential for photosynthesis by which plants synthesize their food. Now in chromoplast, fat soluble carotenoid pigment like carotene, xanthophyll are also present. Carotenoid gives the part of the plant a yellow, orange and a red color. Now the third category is leucoplast. They are colorless plastics with stored nutrients. Sorry, I haven't mentioned here all the three chloroplasts, but I am explaining. Now come to amyloplast. They store carbohydrate, for example in potato. And then comes elioplast, that store oils and fats. Whereas aleuroplast, they store proteins. This is a diagrammatic representation of a chloroplast or the sectional view of the chloroplast. Majority of the chloroplasts are found in mesophyll cells. These are the mesophyll cells. These are the mesophyll cells. Mesophyll cells are lens-shaped, oval, discoidal in shape. Number of mesophylls varies from 1 per cell of the chlamydomonas. In green algae, they are about 20 to 40 per cell in the mesophyll. Like mitochondria, the chloroplast is also double layered, as I have just told you, and of the two, inner chloroplast membrane is relatively less permeable and the space limited by the inner membrane is called as stroma. This fluid is stroma. Now you can see number of organized flattened membranous structure. These structures are known as thylakoids, which is present in stroma. All these thylakoids are arranged in stacks like a pile of coins called grana. Okay, these are grana. Stroma lamella is the name of a flat structure. See, this is the flat structure which connects two thylakoids. Stroma of chloroplast contains enzyme for the synthesis of carbohydrate and protein. Like mitochondria, chloroplast contains small double-stranded circular DNA and ribosomes in it. See, this is chloroplast. Now come to cytoskeleton. Cytoskeleton, it helps cell to maintain shape, move, and they move cell organelles. Okay, they are of three categories. One is microtubule, actin filament, intermediate filament, and microfilaments. This is a diagrammatic representation of a cytoskeleton. This is cell membrane or plasma membrane, this blue part, and these are the endoplasmic reticulum, and these are the microtubule, which is the first part of a cytoskeleton. This is a mitochondria. This is again a microtubule. This is again a microtubule. Okay, and these are the microfilaments and the intermediate filament for providing support. Okay, and these are the ribosomes. These granular structures are ribosomes. Now come to microfilament. Microfilament are fine thread-like protein fibers. They are about 3 to 6 nanometer in diameter. They are composed predominantly of a contractile protein called actin, which is the most abundant cellular protein, which is also present in the muscles. Microfilaments association with the protein myosin is responsible for muscle contraction. And microfilaments can also carry out cellular movement, including gliding, contraction. Then comes the second part of a cytoskeleton, that is intermediate fiber. Intermediate filaments are about 10 nanometer diameter and they provide very hard strength for the cell. Now comes the third category of a cytoskeleton, that is microtubule. Microtubule, they are cylindrical tubes. They are about 20 to 25 nanometer in diameter. They are composed of subunits of a protein, tubulin. These subunits are termed as alpha and beta. Microtubules, they act as a scaffold to determine cell shape and provide a set of tracts for cell organelle and vesicle to move on. Microtubules also form the spindle fibers for separating chromosome during mitosis. When arranged in geometric patterns inside flagella and cilia, they are used for locomotion. So we will discuss cilia and flagella in detail. Cilia. It is a cellular organelle made of exonemal microtubules. It is a specialized region of the cell membrane or surface. These appendages are used for locomotion, for moving from one place to another, and second is the movement of the fluid over the cell. There are two types of cilia. One is motile cilia that helps in the movement, and non-motile cilia which does not move at all. Motile cilia constantly beat in a single direction. They always beat in single direction. Whilst, or on the contrary to this, non-motile cilia typically sense the cell's environment. Now comes the flagella. Flagella are relatively long tail-like or whip-like structure that are capable of either undulating or rotational movement. They are like hunter. Now this is a distinguishing feature of cilia and flagella. Cilia is 0.25 mm in diameter, which is about 250 nanometer, and it is in length, it is about 2 to 10 millimeter, and they are present many per cell, many on a single cell. For example, ciliated protozoa and ciliated epithelia of human cell. And now comes the flagella. It is about 0.5 millimeter in diameter, same as that of cilia, but its length is 10 to 1200 millimeter, whereas cilia is just 2 to 10 millimeter in length, and it is usually present one on one cell. Okay, for example, sperm flagella. Thank you. This comes to an end, and in next class we'll be discussing about nucleus and chromosomes. Thank you for watching Edupedia Word videos.